Hello everyone, we're going to be talking about gene structure in eukaryotes, and we're going to be talking about them in eukaryotes because we're going to be talking about the process of transcription and translation and trying to understand, what are the ways that we can identify gene structure, what are the elements within gene structure, and basically trying to help you, put this into a real life scenario. So we'll look over some real examples as well. So here's kind of just a very simple schematic I've put up, looking at the different levels of transcription and translation. And we're going to work our way through top down, left to right to try to figure out what are these elements, try to identify what's going on. So the first thing is I want to point out that this blue region is referring to DNA. So you can see that if you have high enough resolution, we've got our double-stranded DNA. And then we usually use large boxes to identify important elements in a gene structure. So the first region that's going to be highlighted in yellow are going to be referring to regulatory elements. So these are going to be regions that help to regulate important expression of the genes. So this might be in response to development of maybe the cell, development of the organism. Maybe it might be in response to environment. We might see a change in expression due to the expression of other genes. So there's lots of intense networks. So I'm just going to put regulatory regions. And these are often, they can be very high upstream, they can be close, or they can be very far downstream. So here's another region that's regulatory. Now, when we get really close, we're actually referring to a region called the promoter, and the promoter is where the, close direct elements of expression will be found, as well as where the RNA polymerase is going to bind. So it's going to bind close to this area. We can have a region very close that we call the core, or a region kind of far away that we call proximal. And this is where RNA polymerase is actually going to bind. Now, when we get into the actual more of the specific gene, we're going to have regions that are on either side that are not translated. They'll be transcribed, but these are called UTRs, so untranslated region. And we often will say, because on the left side here is our 5' and on the right is 3' because we're moving 5' to 3', left to right. We'll call this left side the 5' UTR, and the right side is called the 3' UTR. You'll see that later on it'll be translated because the start of transcription happens right at the beginning of the UTR, but the start codon for the translation is found right here. So I'm going to put start. And over here, we have our stop codon. Okay, so then that leads us to these big red boxes here. These red boxes are referring to exons. Exons are going to be regions that actually lead to protein coding regions. So if you want to try to think about it, I always think of exons are expressed, whereas introns, which are these white regions, are spliced out. So they're going to go through processing for the mRNA, which is going to be the next step and what's going on. So in diagrams, often we like to, made these introns these very large blocks, but often they're just lines, like just to really emphasize where the exons are and kind of help show you what's going on. You'll see that when we go through some real examples. So now what's going to happen is this is going to be transcribed. We're going to go down. Now in this region, we're going to be dealing with RNA. You can see we still have our UTR that was transcribed. We still have our start codons. That's going to be necessary for translation. We still have our exons. We still have our introns. What's going to happen is we have these little, these borders between the exon and intron right here. And there's some sequences here, along here, sometimes right in the middle of the intron, that help to identify where we're going to be splicing out the actual introns. What's going to happen is we're going to remove them all together, and they're going to be cut out. Then we're going to be adding, for stability, we add a poly A tail to the end, and we're going to be adding a G cap to the front. This helps for transport outside of the nucleus because that's where we're going to be getting, that's where the messenger RNA is actually going to transport to the ribosomes for translation. So GCAP is on the 5' end. That's going to be referring to guanine, and then polyatyl adenine, that's going to be our polya tail. So I've got some little diagram references there. Now we have our mRNA. Whoop, there we go. Okay, now what's next is we're going to actually, this mRNA protein is going to travel and lead to, sorry, not mRNA protein, mRNA molecule is going to transport outside the nucleus, and we're going to form a protein. So what's going to happen is when it enters the ribosome at the start codon, we're going to read and form our polypeptide. I'm just going to do a little skinny thing here. I'm going to do little circles to indicate that this is a polypeptide that will then, in this case, it'll, with some chaperone proteins, fold in on itself, and then there we go, it's done. It'll go do its work or its job, whatever its role is. So let's just kind of, little summary. We have, a sequence at the beginning to help identify when transcription starts. We have a terminator, which I forgot to mention, at the very end, that helps to stop the transcription. We also have our start and stop sequences to help identify where the codons, the actual coding region is. That is where the UTRs will no longer be found in the protein. They're kind of ghosts. And then we have our protein. So this is how we identify proteins, how we like to, represent genes as structures. And let's go over some real world examples. Okay, so now we're looking at some real life examples. So what I did was I just looked in the BRCA1 gene in NCBI, which is a National Center for Biotechnology Information. It's a very commonly used website for any information on genes, for proteins. It's usually kind of summaries, and it's kind of like a place to put all the information that researchers collect. So if I'm just going to scroll down here, you can see we've got our genomic sequence. And so this is our BRCA1 gene, and we can zoom in here. And these blocks here, like this, is referring to exons. We can actually open this up, and the green regions refer to exons, and we can go to a legend here. You can see that purple is in reference to RNA, red regions here are in reference to coding regions. So the little blocks of these exons, they're kind of hard to see, and that's because these introns are so large. So this is kind of a real world example. But we can zoom out. We can even move along the genome, and we can just look at another gene. Like here, we've got another gene right here. We've got some exons, which are here. The blocks, they're represented in green. I'm going to close this just so we can see more detail. And you can see we have directions of the exons and the introns. There's arrows going left. So remember, DNA is double-stranded. We read along 5' to 3'. So even though we're reading right to left, we're actually reading the opposite strand of if we were, reading in the direction that the genome is listed here. So genes can be, they can be running in opposite direction to each other. If we compared to, here's another gene running right to left in this orientation, but the BRCA gene is actually reading left to right. So here's one example. I really suggest just look up any gene, just look up maybe popular genes, top genes in humans, and then you can look at the graphical representation of what's going on and try to see what are some common elements between them. So there's lots of information that's provided and lots that you can look through. Here's another example. This is the gene that I studied for my thesis, actually, for my master's, and you can see that there's 3 splice variations, so they have roughly the same exons and introns. There's just slight differences in them that are found when we actually look at the mRNA, so they are spliced in a different variation. So if I click on one of them, we can actually look at information regarding this is in plants. So in plants, we have a little simpler diagram for our genes. You can see there's some others here. So these are the genes, the regions, the red areas, and then the splice variants are found down here showing the actual gene structure. And then very common mutation points down here at the bottom, where you can maybe order even plants that have been mutated, specific seeds for these variants. Okay, so if I click into one of them, we can go down, and here's the sequence, which is found. So we could do some bioinformatic work if we wanted. If we go all the way to the bottom, we can see, look at this, ATG, there's our start sequence of our coding region. So very interesting, very cool. And with that, I want to thank everyone for watching. Don't forget to subscribe, it really helps the channel. And hopefully this will help in your schooling or research or whatever you're doing. So all the best.